Good morning, class.
Good morning, Teacher Del Monte.
So, how's your day? So, for today's discussion, we are going to tackle about a chemical name for a molecule that carries genetic instruction in living things, which is the deoxyribonucleic acid, or what we also call the DNA. Okay, so before anything else, let me ask someone here who can share their ideas about or when it comes to DNA. Okay, yes, Kulas. Okay, can we hear your thoughts?
Ma'am, idea about DNA. For me, DNA forms a shape known as double helix.
Okay, as we commonly see that the DNA structure is somewhat a double helix form. Another thing to know about DNA is that it associated with the transmission of genetic information. Okay, so now let's go beyond the DNA and let's talk about the synthesis of DNA, which is the replication. Okay, so now what do you mean by DNA replication?
There are 4 steps in replication of DNA. Step 1 is the replication fork formation. Before DNA can be replicated, the double-stranded molecule must be unzipped into 2 single strands. DNA has 4 bases, first called adenine, second thymine, third cytosine, fourth guanine, that form pairs between the 2 strands. Adenine only pairs with thymine and cytosine only binds with guanine. In order to unwind DNA, these interactions between base pairs must be broken. This is performed by an enzyme known as DNA helicase. DNA helicase disrupts the hydrogen bonding between base pairs to separate the strands into Y shape known as the replication fork. This area will be the template for replication to begin. Step number 2 is the primer binding. The leading strand is the simplest to replicate. Once the DNA strands have been separated, a short piece of RNA called a primer binds to the tree end of the strand. The primer always binds as the starting point for replication. Primers are generated by the enzyme DNA primase. Step number 3 is the elongation. Enzymes known as DNA polymerase are responsible creating the new strand by a process called elongation. The logging strand begins replication by binding with multiple primers. Each primer is only several bases apart. DNA polymerase then adds pieces of DNA called Okazaki fragments to the strand between primers. This process of replication is discontinuous as the newly created fragments are disjointed. The 4th step in replication of DNA is the termination. Once both the continuous and discontinuous strands are formed, an enzyme called exonuclease removes all RNA primers from the original strands. These primers are then replaced by appropriate bases. Another exonuclease proofreads the newly formed DNA to check, remove, and replace any errors. Once completed, the parent strand and its complementary DNA strand coils into the familiar double helix shape. In the end, replication produces two DNA molecules, each with one strand from the parent molecule and one new strand.
So that's it, okay? So those are some of the steps in which DNA replicates. Okay, so is there any question, clarification, or volcanic eruption? Shut up.
Not so far, ma'am.
Actually, we only have one topic left, which is the DNA translation. So since it's already time, and we still have class tomorrow, so for tomorrow, we will have only a reporting with the topic of DNA translation. So the scheduled reporters for tomorrow is Cordapia and Procolo. Okay? So class, please make some advanced studies about our topic for tomorrow's reporting.
Okay, ma'am.
Okay, so this ends our class. Okay, so thank you and goodbye.
Goodbye and thank you, Teacher Del Monte.
Hi class, good morning.
Good morning, Teacher Del Monte.
So let us pray in the name of the Father, the Son, and the Holy Spirit. Amen. So yesterday we talked about the DNA replication. So as what I've said, today we will have our reporting with regards to the topic of DNA translation. So reporters, are you both ready now?
Okay, ma'am.
Hi, guys. So, now we are going to explain and discuss a kind of synthesis in genetic transmission, which is the DNA translation. But before we start, may I ask, Perlita, how does DNA translate? Perlita, please stand up.
Based on my own understanding, ma'am, in order for a DNA to translate, it takes process or steps for the final translation.
Okay, thank you. Well, according to Perlita, it has the following steps in order for the DNA to translate. But actually, the process or steps will be explained later by Procolo. So now, let me explain first some background and informations about the DNA translation. Translation is the process of translating the sequence of a messenger RNA molecule to a sequence of amino acids during protein synthesis. The genetic code describes the relationship between the sequence of a base pairs in a gene and a corresponding amino acid sequence that it encodes.
Hi guys, so after knowing the definition and information of DNA translation, so now I'm gonna explain to you the process and how does DNA translate. First step, initiation. For translation to begin, the start codon 5AUG must be recognized. This is a codon specific to amino acid methionine, which is nearly always the first amino acid in a polypeptide chain. At the 5 cap of mRNA, the small 40S subunit of the ribosome binds. Subsequently, the larger 60S subunit binds to complete the initiation complex. The next step can now commence. Second step, elongation. The ribosome has two tRNA binding sites, the P site which holds the peptide chain and the A site which accepts the tRNA. The ribosome then translocates along the mRNA molecule to the next codon, again using energy yielded from the hydrolysis of GTP. Now, the growing peptide lies at the P site and A site is open for the binding of the next amino acid tRNA, and the cycle continues. The polypeptide chain is built up in the direction from the N terminal, which is methionine, to the C terminal, which is the final amino acid. Third step, termination. One of the three-step codons enters the A site. No tRNA molecules bind to these codons, so the peptide and tRNA in the P site become hydrolyzed, releasing the polypeptide into the cytoplasm. The small and large subunits of the ribosome dissociate, ready for the next round of translation. Okay, so those are the three steps in a DNA translation. First is initiation, second is elongation, and the third one is termination. After these processes, the DNA can be formed or ready for a new translation. So I hope you understand it all. Thank you and God bless.
Okay, so let's give a clap to Procolo and Cordapia. So thank you both for a well-explained reporting. Okay, so is there any question, class, or clarification regarding with the said topic?
None so far, ma'am.
None, ma'am.
So I think that would be all for today since it's already time. So goodbye and thank you, class.
Goodbye and thank you, Teacher Del Monte.